Hi class, welcome to our topic cell transport. So in this video, we will discuss how molecules move through cell membranes. Okay? So how molecules move through cell membranes? We will answer these questions. Where can you find membranes in the cell? What are the functions of the cell membrane, and what is a semi-permeable membrane? So our first question is, where can you find membranes in a cell? As a recap of our previous lesson on the different organelles or the different parts of the plant and animal cells, we discussed the membranes. So membranes here in our illustration is found in the various parts of the cell. Different organelles have also membranes, or they are called membrane-bound organelles. So the main membrane is the cell's membrane, which surrounds the whole cell. So here in our illustration, we can see the cell membrane that protects the cell from the external environment. It encloses the cell. Additionally, there are membranes inside the cell called organelle membranes, such as what? We have the nuclear membrane that surrounds the nucleus of the cell, the endoplasmic reticulum, mitochondria have its membranes. So these membranes help separate different parts of the cell and perform different functions. So next, what are the functions of the cell membrane? What is the role of the cell membrane? The cell membrane acts as a selective barrier. It's a barrier, just like having control guards. So when you enter or exit a restricted area or any establishment like banks or malls, so you can see the guards, security guards, they restrict, or they are the one who controls the persons who will enter or exit a certain area. So just like the cell membrane, it acts as a selective barrier that controls the passage of substances in and out of the cell. It also regulates the entry and exit molecules and ions, allowing only certain molecules to pass through while blocking others. Next one is the functions. So what are the functions of cell membrane? Cell membrane acts as what are the different functions? So the main functions of the cell membranes is it's selective barrier. However, there are also equally important functions of the cell membrane, and it is stated here in our slide. The cell boundary, it regulates the materials and maintain homeostasis. So what are these? So the cell have important function, and one of it is its protective barrier, enclosing the cell and keeping harmful substances out. It controls the entry and exit of molecules and ions, regulating what goes in and out of the cell. Also, the cell enables to communicate with each other and their environment through receptors and signaling molecules. It helps maintain the cell's internal environment by controlling the balance of ions and molecules inside and outside of the cell. And lastly, The cell membrane provides structural support of the cell. All in all, it maintains the balance or homeostasis in the cell. So what is a semi-permeable membrane? Okay, membranes that allow certain materials to pass through based on certain properties, size, hydrophobicity, and charge. So it is semi-permeable because it will allow some, not all, of the materials. A semi-permeable membrane is like a filter that allows substances to pass through while blocking others. As you can see in the picture, it is like strainer or sieve that will filter some of the substances. It's semi-permeable because it permits the passage of certain molecules or ions based on their size, charge, and other properties. For example, a cell membrane is semi-permeable because it lets certain molecules like water and small ions pass through while blocking larger molecules like proteins. This selective permeability is essential for controlling what enters and exits the cell. Okay, here is in the illustration, you can see the fluid mosaic model. So it is a model. We are saying that the cell membrane is made up of tiny pieces that can move around like they're floating in a liquid. So that is why it is called a fluid mosaic model. It is called fluid because these pieces can shift and change position, making the membrane flexible. So you will think the cell membrane as a flexible structure made up of small parts that can move around like tiles in a mosaic floating in a liquid. Okay? Next one are the phospholipids and proteins make up most of the membrane. So in the cell membrane, most of it is made up of phospholipids and proteins. So cholesterol is there to make it flexible. Like how a bit of oil helps make dough stretchy. So here we have a lipid bilayer. So cholesterol. And there are carbohydrate chains. So we can see here carbohydrate chains that help the cell talk to other cells. Some like it is like you can see here, you can visualize that it is like an antenna or like a secret code for communication. It's like a team of phospholipids and proteins with some flexibility from cholesterol and a secret language from carbohydrate chains that makes up the cell membrane. Okay? So here is a picture of a phospholipid. Phospholipid is like tiny building block in cell membrane. It has two tails. These are the tails of a phospholipid that is made up of what? Fat. And the head contains phosphate. Okay? So it's like a small structure with two fatty tails and a phosphate head. So the phosphate head of the phospholipid is like a friend who loves water and wants to near it all the time. So that is why it is called hydrophilic or water loving. However, on the other side, we have the fatty acid tails. So these fatty acid tails are like friends who don't like water, kandingon, and all then try to stay away from it. So it is called hydrophobic. You have a phobia with water fearing. So in phospholipid, one part is hydrophilic and the other one is hydrophobic. Okay? So that is the definition of hydrophobic and hydrophilic. Hydrophilic tails and hydrophobic heads. Next is the phospholipid bilayer in a cell. Phospholipid bilayer forms because the inside and outside of the cell are mostly water. So, water, hydrophilic. Yan. Okay? So, you can see here, the inside is water and the outside of water. So, it is a bilayer, 2 layers. Here in the inside, we have the heads turning inwards, while outside, the heads are turning outwards. And the center are those hydrophobic tails. Okay? So, the phospholipid bilayer is a semi-permeable membrane because it only allows certain molecules to cross. So, as we discussed earlier, the phospholipid bilayer has 2 layers of special fats. The heads of these fats are hydrophilic, while the tails repel water, hydrophobic. So, this structure creates a barrier. Mura siya og wall. A very hard wall. Now, some molecules, like small ones and those that are also hydrophilic or water-loving, can easily pass through the phospholipid bilayer because they are compatible with the outer hydrophilic heads. So, those water molecules can freely move from the outside to the inside, inside to the outside, because they are water-loving. Okay? So, we have here why this happens. It is semi-permeable. This property helps the cell control what enters and what exits, maintaining a controlled internal environment. So, we have here hydrophobic and polar. Large and charged molecules use a transport protein to enter and exit the cell. So, this is the image of a transport protein. Okay, so hydrophilic and polar, these molecules are like water and have a positive and negative end. So, they are like magnets for water. Large and charged means that some molecules are big and carry an electric charge. They have an electric tag. Okay? So, some molecules like big, charged, and water-loving ones can squeeze through the gate in their own because they are too special. So, mga special, pwede ra makapass through. To get to the gate, they need a special escort like a VIP pass. These escorts are the transport proteins. So, these transport proteins are very picky. So, pilian kayo ni siya. Pareha sa imong gipanguyaban. Pilian kayo. So, each one only lets a specific type of molecules through. It's like having a key that only fits one lock. So, specific. It's very specific. So, example. Let us have an example. So, example of transport protein exclusive for glucose molecules. So, may paning mga transport proteins, no? Kuan kaya sila? They are like loyal. Di sila kabalo mga bit. So, they are loyal to the one they chose. So, if the transport protein is exclusive for glucose molecules, so glucose molecules lang ang i-allow niya to pass through. The same with other transport protein, exclusive for other molecules. It won't let anything else through but glucose. Okay? So if you're a big charged water-loving molecule like glucose and you want to enter and exit the cell, you have to find the right transport protein at the gate mo. Nandili ka magmadali. Kay na-adjud si Mr. Right. Okay? Naadjud nakalaan para sa imuha. Naadjud kay pair. Naadjud best friend nga gilaan si Lord sa imuha. Your better half. So, dili lang ka magdali if ever naka-crush ka or you are in love with a person and it is just one-sided. Ikaw lang ang nakagusto sa iya, niya siya, wala nakagusto sa imuha, lahi ang gusto. So, don't be sad. Naadjay para sa imuha. There's one perfect for you. O pareha sa transport proteins o ang transport proteins gani naadjay gilaan nga molecule para sa iya ha. So ang buhaton lang atong nga molecule pangitaon niya asa ang para sa iya ha. Asa siya nga transport protein pwede maka-pass through. Okay? It's like finding the right key for you. It's like finding the right person for you. So these transport proteins ensure that only the right molecules can pass in and out of the cell. O, diba? Si transport protein manggani makakita manggani the right molecule for the proteins. Ikaw pa kaya? Okay? So next. So we have here protein channel. It is like a tunnel through the cell membrane. So this tunnel is like a superhighway specifically designing for big water-loving and charged ions. Okay? Okay. So wala na siya kinahanglan nga energy. A special entry for large hydrophilic charge diffused through the cell membrane. It is called facilitated diffusion. So they are just sliding down from high concentration to low concentration. It is called facilitated diffusion. Okay, it's like the tunnel that makes it easy for these ions to go through where they need to go without using a lot of energy, just like a fun slide at the playground. So unlike this kind of sodium-potassium pump, wherein you really need to exert energy to move these molecules from other side. Okay? So, as a summary, we will now watch a video about cell membrane.
In my second year of teaching, I was looking to improve an osmosis lab I had done the year previously. Osmosis, if you remember from our osmosis video, involves water traveling through a semi-permeable membrane, potentially a cell membrane. And I wanted a cool way that students could model this in different scenarios. And one of my colleagues told me about this egg lab. I won't get into the whole lab though. It was actually one of the first steps of the procedure that got to me. To get ready for the lab, my colleague told me, you can soak eggs in vinegar for 24 to 48 hours and the shell comes off. Oh, so I need to make hard-boiled eggs then. No, raw eggs. But if the shell comes off, well, that's the whole point. What's underneath the shell is going to mimic a cell membrane. It's kind of modeling how a cell membrane would function, you know, if the whole egg was actually like a cell. So then you run different scenarios with it for your osmosis lab because it will be a semi-permeable membrane, like a cell membrane. I couldn't visualize this. If the shell comes off, but it's a raw egg, how does it stay together? So I have this area in my house that's designated for things for me to try out. Don't worry, I always clean up afterwards. I tried this experiment out in advance. just to be sure. The hard shell is removed, but the membrane that was always there remains. We often visualize the membrane of a cell this way, like this membrane around the chicken egg. The cell membrane is semi-permeable, meaning it lets some materials through, but not others. We have an entire video all about cell transport and how materials can pass through the membrane. A cell could never be as large as a single chicken egg, though. Why? Well, it turns out surface area is a very important thing. Remember that surface area determines the surface measurements of that cell membrane, and the cell membrane controls what goes in and out of cells. That includes food coming in, as well as molecules that are essential for metabolic processes, and then also waste going out. If volume, which is all this space inside, increases, then you will have more need for surface area, as you will have more need for food to enter, more of a need for waste to be removed, and more metabolic reactions occurring in this larger volume in the first place. If we do a little bit of math here between these two models, I'm going to use popular cube models instead of egg-shaped models because it's a little faster for me to do surface area and volume calculations. See how here there is a big difference in surface area to volume in this smaller model? 6-to-1 ratio. That means the surface area in this small model is 6 times more than the volume. Look at this beautiful ratio with so much surface area. But if we look at this bigger cube and we do some math for this model, that surface area to volume ratio decreases. Sure, the surface area is still larger than the volume in the large model, but it's only 2 times as large now, not 6 times as large. Cells are way smaller than this small model here to allow for an exceptionally large surface area to volume ratio. And a major reason why we're not going to find a cell as big as this chicken egg here. Surface area is important. And while we can model a lot of the processes of cell transport from this egg membrane and how important the membrane is, I don't want to neglect talking about the amazing cell membrane structure itself, because the cell membrane structure truly is magnificent. And since every single living thing is made up of one or more cells, which is part of the cell theory, it's a big deal because every single cell has a membrane. It doesn't matter whether you're talking about bacteria, or protists, or plants, or animals, or fungi. Even archaea aren't too cool to have a membrane. They all have a cell membrane. The structure can vary some, but we're going to be talking about some major structures of the membrane that you can actually find in most cells. We should mention that the fluid mosaic model is often what we use to describe the cell membrane. A mosaic, in case you've ever created one, we did in some of our art classes over time, arranges many small pieces together to make some large piece. You'll see that that makes sense when describing the membrane in a minute. The word fluid implies movement, and this is true for the cell membrane as the components are floating around, they're not static. So let's take a look at some of these components in the cell membrane. We're looking first at a phospholipid bilayer. A phospholipid is a lipid, but an interesting one. So when you talk about a lipid in general, many lipids are nonpolar. Think of oil, for example. It's nonpolar. It won't dissolve in water. Water is polar. But a phospholipid is interesting because one part of it is polar, the head, and the other part of it is nonpolar, the tail. It's amphiphilic. Let's explain what we mean. We often refer to the polar head of the phospholipid as hydrophilic, which means that part loves water. Well, you know, if a lipid could love. The nonpolar tails are hydrophobic. They do not like water. These phospholipids arrange themselves into a phospholipid bilayer with the nonpolar areas here in between, away from any water. It also allows this area in between to be separated from the outside and inside. Water can be found on the inside and outside areas. Also, these phospholipids, they don't just stay put. They move around. It's the fluid mosaic model after all. This gives the cell membrane flexibility. Phospholipids can even flip-flop around, but that's far less common. Remember that this entire phospholipid bilayer borders the whole cell. It would be a sphere, even though we're just looking at one area of it. We have an entire video that talks about which molecules can go in and out of this membrane and which ones can't that you can view. But for now, we're going to look at some of the other structures more in depth. Cholesterol. You know, cholesterol often gets a bad reputation. And while cholesterol that builds up in the arteries can be a problem, cholesterol in your cell membrane is critical. If temperatures drop, the cholesterol can actually function kind of like spacers between these phospholipids, keeping them from becoming too packed. Or vice versa, the cholesterol can actually function to connect phospholipids to keep them from becoming too fluid in warm temperatures. Proteins. In protein synthesis, we talk about why it's so important for cells to make proteins. Many proteins are found in or on the cell membrane, and they play major roles. Peripheral proteins, like the name suggests, tend to be on the peripheral area of the membrane. So while they're on exterior areas of the membrane, they generally are not going to go through the membrane. That's for integral proteins. Integral proteins go through the membrane. Oh, and peripheral proteins can sit on them sometimes. Because of location, these proteins tend to have different functions. Integral proteins, with their potential to go through the membrane, are frequently involved in all kinds of transporting methods for all kinds of materials. Some relevance? Consider the breakfast you ate this morning. Your body digested what you ate for breakfast to obtain glucose. Once in the bloodstream, those glucose molecules can't just squeeze through the phospholipid bilayer to enter all of your cells. The glucose molecules are too big and polar. But your cells need glucose to survive, to make ATP, and they rely on integral proteins to get it. Peripheral proteins tend to be more loosely attached, since they're generally not stuck in the membrane. They have an assortment of functions, such as acting as enzymes to speed up reactions, or attaching to the cytoskeleton structures to help with cell shape. Both protein types can have carbohydrates bound to them, which can make them considered a glycoprotein. If the carbohydrates attach to a phospholipid, you could have what's called a glycolipid. Glycoproteins and glycolipids can identify the cell as belonging to the organism, self and non-self recognition, which is very important when you're fighting pathogens. They can also be involved in many kinds of cell signaling. In fact, here's some relevance. A glycoprotein known as CD4 is found on the surface of some of your immune cells. The CD4 glycoprotein is essential for some of these immune system cells to interact with each other and activate. However, it's also exploited by the HIV virus. The HIV virus uses that CD4 glycoprotein as a way to bind to helper T cells, which it then can infect. Understanding the components of the cell membrane and how those components are involved in recognition and cell signaling is critical to understanding how to fight back against many viral and bacterial diseases. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so that is the end of the discussion on the cell membrane. So we are going to now proceed with the active and passive transport. So, which among these pictures describe a person who is passive? Are you a passive student or you are an active student? Okay, so here is the person who are not moving, relaxing. So it is a passive one, while this picture of person who is biking is an active person. Okay? So here is the definition of a passive transport. Materials move down the concentration gradient from high to low solution concentration. So it is like you are biking downhill. So even if you are not pedaling, you are not exerting effort because of the force of gravity, your bicycle will move from the top of the hill and down the hill. Okay? Equilibrium is reached so when there is an equal concentrations. No energy is needed by the cell to move the materials because you are relying to the concentration from high to low concentration until it will reach the equilibrium or pantay na sila. Next one, the contrast of active transport is of the passive transport is the active transport. Here you can move materials even from low concentration to high concentration. with, of course, the expense of energy. So you can only move materials in an active transport if you will use ATP or you will use energy. So like this illustration, so you can see if you really want to climb up the hill with your bicycle, you really need to exert much effort to climb the hill. So that is active transport. So there are two main ways that materials can move in and out of the cell. We have the passive transport and active transport. Under these transport mechanisms, we have different types of transport. So passive transport, we have diffusion, facilitated diffusion, and osmosis. While the active transport, we have protein pumps, endocytosis, and exocytosis. Okay, we are already done with this diffusion and osmosis from high concentration to low concentration, and we also have proteins to facilitate the diffusion. So here we have in the active transport, we have protein pumps. So it uses energy to increase the concentration on the other side of the membrane. We also have this example, the sodium-potassium pump, an example of protein pumps. Uses ATP to move Na, so Na, this is sodium, out, and K, or the potassium, into the nerve, muscle, and many other cells. Okay? So what is the job? The job is to move the sodium out and bring the potassium in. Okay? It is important for the cell to function properly. So now the energy source is the ATP, adenosine triphosphate. It's like a battery. And we have the machine that pumps the potassium and sodium, the protein pump. Okay? The sodium potassium pump is like ball exchanging machine that uses ATP to pump out the sodium and bring in the potassium, ensuring the cell's proper function, especially in what? The nerve and muscle cells. Here is another application of protein pumps. You can observe this one during photosynthesis. So in photosynthesis, plants and some other organisms use energy from sunlight to convert carbon dioxide and water into glucose and oxygen. So this process occurs in the special structures called the chloroplasts. So the protein pumps here, these are the pumps. These are the protein pumps that actively transport ions like hydrogen ions across the membrane. These pumps use energy from ATP to create a concentration gradient. So from this one, from this thylakoid space, it will transport hydrogen ions to the stroma. So if there are lots of hydrogen ions here, Thus, it will create a concentration gradient of hydrogen ions. So, it will make the ATP synthase spin and generate ATP. Okay? So, these are the pumps, protein pumps. We will have a thorough discussion of these topics in the second grading. Next one is the endocytosis. Endo. So look, as you can see in the illustration, it brings materials from the outside of the cell into the cell. Okay? It is like wrapping up these molecules. So the outer membrane of the cell is a very flexible material that it can carry, or it can bring something in, like big molecules or even tiny organism. So it starts wrapping, making curve, and then wrap the materials in its membrane. Then it forms vesicle or creates bubble inwards, and it eventually closes the materials completely, forming a bubble-like structure inside the cell. So these bubbles are called vacuole. Okay? So now the materials are inside the vacuole. The cell can safely transport it into where it is needed or store it for later use. Okay? It needs to eat up harmful bacteria or debris. So in the case of white blood cells, the white blood cells uses endocytosis. So it wraps its membranes around bacteria or debris, creating a vacuole inside itself. Then, special enzymes inside the cell can break down the bacteria, helping the white blood cells get rid of the threat. So, endocytosis, it's like the cell's way of packaging and dealing with the unwanted items or getting essential nutrients without letting them directly touch its insides. Okay? So, exocytosis, kabaliktaran ng endocytosis. Endo, inside. Exo, outside. So, kanina, vacuole ang na-form. Sa exocytosis, the vesicles fuse to the cell's membrane to release their contents, the cell products or waste. So, as you can see here, it secretes the unwanted materials or waste, or it will give out its cell product. Okay? So here is the animation of how the phospholipids form vesicle membranes, integrate into the plasma membrane to get rid of those material. Okay? So here, I prepared a video animation about phospholipids, or about the overall cellular transport mechanisms, as a summary of the things we discussed about cellular transport mechanisms.
Have you ever wondered what it might be like to be inside a cell? Imagine the genetic material, the cytoplasm, the ribosomes. You'll find those in almost all cells, prokaryotes and eukaryotes. Eukaryote cells, in addition, have membrane-bound organelles. All of those structures and organelles have different functions, but cells are not isolated little worlds. They do have a lot going on inside them, but they also need to interact with their environment. It makes sense that to keep a stable environment inside themselves, otherwise known as keeping homeostasis, they must have some control on what goes in and what goes out of them. A very important structure for this that all cells contain is the cell membrane. By controlling what goes in and out, the cell membrane helps regulate homeostasis. It's the homeostasis king. Let's take a look at the cell membrane. You could have a whole course on the cell membrane itself. It has amazing structure. It has signaling abilities. But to stick to the very basics, it's made of a phospholipid bilayer. Bilayer means two layers, so you have these two layers of lipids. Now, these lipids, they're called phospholipids, well, they have a head that is polar, and they have a tail that is nonpolar, making them quite unique. Some molecules, they have no problem going through the cell membrane, and they directly go through the phospholipid bilayer. Very small, nonpolar molecules, they fit in this category. They're a great example. Like some gases, oxygen and carbon dioxide gas, those are great examples. This is known as simple diffusion. Also, it doesn't take any energy to force those molecules in or out, so it's known as passive transport. Simple diffusion moves with the flow, meaning it moves with the concentration gradient. Molecules move from high concentration to low concentration. So when you hear someone saying that something's going with the concentration gradient, that's what they mean. They mean it's going from a high concentration of molecules to a low concentration of molecules. Now, remember how we said the cell membrane is actually a pretty complex structure? Well, one thing we haven't mentioned yet are proteins in the membrane, and some of them are transport proteins. Some transport proteins act as channels. Some of these proteins actually change their shape to get things across. Some of them open and closed based on some kind of stimulus. All of these are good things because it's helping with molecules that may be too big to cross the membrane on their own, or molecules that are polar and therefore need the help of a transport protein. This is known as facilitated diffusion. It's still diffusion, and it still moves with the concentration gradient of high to low. It doesn't require energy though, so it's also a type of passive transport. It's just the proteins are facilitating or helping things pass. Charged ions often require a protein channel in order to pass through. Glucose needs the help of a transport protein to pass through. In osmosis, for water to travel at a fast rate across the membrane, it passes through protein channels called aquaporins. So these are all examples of facilitated diffusion, which is a type of passive transport and moves with the concentration gradient of high to low concentration. Now all the transport we've mentioned has been passive in nature. That means it's going from high concentration to low concentration. But what if you want to go the other way? For example, the cells lining your gut, they need to take in glucose, but what if the concentration of glucose in the cell is higher than the amount of glucose concentration in the environment? We would still need to get the glucose in, so it's going to have to be forced against the regular gradient flow. Movement of molecules from low to high concentration will take energy because that's against the flow. Typically, it's going to require ATP energy. A reminder that ATP, adenosine triphosphate, it has 3 phosphates, and when the bond for the last phosphate is broken, it releases a great amount of energy. Yeah, so ATP is a pretty awesome little molecule. ATP can power active transport to force molecules to go against their concentration gradient. And one way it can do that is actually energizing the transport protein itself. One of our favorite examples of active transport is the sodium-potassium pump. So that's definitely something worth checking out. There's other times a cell needs to exert energy for transport. We're still in active transport right now, but let's say a cell needs a very large molecule. Let's say a big polysaccharide. If you check out our biomolecule video, that's a large carbohydrate. Well, you may need the cell to fuse with the molecules it's taking in order to bring it inside. And this is called endocytosis. Think endo for in. Often this fusing of substances within the cell membrane will form vesicles that can be taken inside the cell. Endocytosis is kind of a general term. There are different types of endocytosis depending on how that cell is bringing substances inside. Amoebas, for example, rely on a form of endocytosis. In this example, pseudopods stretch out around what they are going to engulf, and then it pulls it into a vacuole. And there are other forms too, such as the fancy receptor-mediated endocytosis. This is where cells can be very, very, very picky on what's coming in because the incoming substances actually have to bind to receptors to even get in. Or pinocytosis, this allows cells to take in fluids. So to the Google to find out more details of the different types of endocytosis. Exocytosis is the reverse direction of endocytosis. Think exo and exit. They sound very similar too. Exocytosis can be used to get rid of cell waste, but it's also really important for getting valuable materials out that the cell has made. Want a cool example? Well, think back to those polysaccharides. Did you know that large carbohydrates are also really important for making plant cell walls? Cell walls are different from cell membranes. All cells have membranes, but not all cells have a wall. But plant cells do. And if you're going to make a cell wall, you're going to need to get those carbohydrates that are produced in the plant cell out of the cell to make the wall. So there's a great example of when you'd need exocytosis right there. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, and that's the end of our topic on cellular transport. May God bless us always.